Okay, so you were tasked with watching lecture 10 as an asynchronous lecture, and that kind of just introduced the sugars, the monosaccharides, the polysaccharides, and now we're going to talk about what the cells do with those macromolecules. And so really what lecture 11 through I think 13 is just taking us through the breakdown and the modification of a glucose molecule all the way until the cell makes tons of ATP, barring oxygen in the cellular environment. So today we're definitely going to cover glycolysis, which happens regardless of oxygen in the cell or not. And then depending on time, we will start pyruvate oxidation. And so we like to call this process aerobic respiration. Why? Because aerobic means using oxygen. And so another phrase that you'll hear me use is aerobic glucose metabolism, because we'll learn later, maybe around lecture 13, that the cell doesn't need glucose, it prefers it because that's going to produce the most ATP. But that's why I differentiate this by saying aerobic glucose metabolism, because it really does specify the molecule that we're using. Okay, and so in this little cartoon here, it's just kind of like a big picture. Try to imagine where we're at in the cell. So here's just your cartoon of a eukaryotic cell with the nucleus in the middle and these little kidney beans that look like the mitochondria, and of course your cytosol. And so if you really zoomed in at the mitochondria, you have this really nice cartoon that shows pretty much where all of these processes take place. And we talked about this yesterday in discussion, where glycolysis, regardless of the cell type, is going to happen in this cytosol, the yellowy part. And it takes one glucose molecule and converts it into pyruvate, not in one chemical reaction, but 10. And that's what we're going to talk about today. And then this pyruvate has to move into, if it's a eukaryotic cell, it has to move into the mitochondrial matrix. And groups yesterday really highlighted the structures of the mitochondria, so we know what the matrix is now. So really this is the place that we're going to talk about today, or the processes that we're talking about, or this region right here. Okay, so I've posted some extra resources for glycolysis for you. If you do have the textbook, the pictures that you'll see, I think, yeah, the pictures that are in my slides today come directly from the text, the molecular biology of this cell. But I've also included some other resources for you. You'll see that it's another textbook. And I secretly found, this is recorded, but oh well, I secretly found this on Pinterest. Somebody posted these to Pinterest. So if you don't have the textbook, don't worry, you definitely have Pinterest, so go there. And there are descriptions for each step, or I should say reaction. Okay, so we can go ahead and get started. So as I mentioned in the first slide, glycolysis happens first no matter what kind of metabolism you do, whether there's oxygen or not. If there's oxygen, we call it aerobic, or if there's no oxygen, we call it anaerobic. And you can see basically that both of them start with glycolysis. And the starting substrate for glycolysis is glucose no matter what, and it's typically going to be in the ring structure shape rather than the straight chain. And no matter what, you're always going to produce two, so this is one molecule of glucose, you're always going to produce two molecules of pyruvate. Not only that, but you also produce two ATP molecules as well as two NADH molecules. Okay, same. Okay, and then the cell has to make a decision. So if there's oxygen present, then the cell undergoes what's known as cellular respiration. So when you hear the word cellular respiration, technically that does not include glycolysis. So what we like to say is that aerobic respiration is glycolysis plus cell respiration. Oh, it's written there. Huh. Whereas anaerobic metabolism of glucose is glycolysis and some sort of fermentation that we briefly introduced yesterday at discussion. Okay, So cellular respiration, the topic of the next couple lectures. After the cell undergoes glycolysis, those pyruvates undergo a process called pyruvate oxidation, and the products of pyruvate oxidation feed into the citric acid cycle. We call it a cycle because these are a collection of chemical reactions as well, and the final product starts the process all over again. Whereas in glycolysis, the final product does not start glycolysis over again. The final product goes somewhere totally different. So that's why we call this a cycle. And some of the byproducts of this cycle, they are used for the electron transport chain and ATP synthesis. And finally, you have your byproducts of carbon dioxide and water. So I haven't added all of the other ATPs that are made because we're going to be talking about them slowly but surely, or maybe fastly but surely. Okay, so an overview of glycolysis. This is really just kind of just to wrap your head around things just a bit. So first things first, there's 10 reactions in glycolysis. Okay? Reactions 1 through 5, they are net energy using reactions. So we call them endergonic. Okay? But what we also like to say is this is our energy investing. So energy investing, right? You're putting energy, you're putting money into a savings account or a CD or wherever, some investment of money, so that hopefully you get more money in return. That's what steps 1 through 5 are. You're using some of the cell energy and you're putting it into this awesome financial opportunity, because you're going to hope, because the chances look good, that you're going to reap more energy than you put in. So there's your energy investing steps. And the types of energy that is used is typically going to be ATP molecules. And so what you'll see is that in these first 5 steps, you actually use 2 molecules of ATP. And you might be thinking, wait a second, isn't the whole goal of this is to make ATP and we're just using it, so we're in the deficit? Yeah, but that's what investment banking is all about. You have to use money to make money. All right, so in steps 6 through 10, these are in general exergonic. The exception is reaction number 8, uses a tiny, tiny little bit of energy, but collectively, reactions 6 through 10 are exergonic, and we call these energy harvesting. Now it's time to go to get the money that you invested some money and now you got some money back. You want to pull that out of your account and go on a trip. Great, energy harvesting. Or you can pull it to the farm fields. You have to put energy into planting all these things, and then when it's harvest season, you harvest all the grapes to make the wine. Perfect. Okay, so we say that it's energy harvesting. Why? Because you're making high energy storing molecules. These are going to be 4 ATP. So not only did you repay back what you put in, but now you have 2 extra. So yes, in theory, you're making 4 ATP, but the net gain is 2 ATP, right? Because this would be -2 plus 4. Not only that, you also make 2 NADH. And we'll learn later, just for now, I'll just say, hey, you're saying the cells don't use NADH. I never heard of that before. They don't use that directly as energy, but 1 NADH provides enough power to the big molecule that does make ATP. It provides enough power, enough energy for that big molecule, which is an enzyme, to make 2 to 3 ATPs, depending on cell type. So 1 NADH is equivalent to 2 or 3 ATPs. So that's why we say NADH is a high energy storing molecule, because when the cell does use NADH, the products are 2 to 3 ATPs. Okay. Okay, so where does this all happen? We know this by now because we've talked about it a few times. And in prokaryotes and eukaryotes, it happens in the cytosol. Please use cytosol, not cytoplasm, because cytoplasm refers to the liquid jelly part and the organelles. So glycolysis is not happening in all the organelles. It's happening that the enzymes are floating around in the cytosol, and it's happening in the cytosol. It's not happening in the structure. Okay, and so what is the starting substrate? We know that it is a molecule of glucose. For plants and animals, and I think even most bacteria, it's going to be a D-glucose. And then we talked about alpha versus beta. So typically for plants and animals, it's going to be in a ring structure. So it's going to be that hexagonal ring. And in bacteria, it happens to be more common that they use the straight chain. So you have your carbons in a chain and you have the hydroxyl sticking off of those. Let's see what else. So when we say these 10 reactions, essentially every single reaction, you're going to have some substrate molecule. So we'll just call it S. Okay? And that substrate gets modified by some enzyme. So we'll call that ENZ. And that enzyme sometimes needs cofactors. So we'll put that in parentheses because not all steps require cofactors. And the result is some product, PR, because I know that I put phosphate groups as a P as well. So PR is product. That's what every 10 steps is. And so what your goal is as a student is to recognize or be able to formally communicate what happens at each step using the key phrases like, instead of saying the substrate binds to the enzyme, that's now a vague term. So instead you're going to say glucose binds to hexokinase and then gets modified to become, and then you'll tell me the product name. And this enzyme requires the cofactor blah, and this cofactor, and you'll tell me what the cofactor provides for the enzyme. Okay, so it's really about just dissecting each reaction and trying to see how the substrate changed. And so that's what we're really going to do today. Let me make sure that everything that I wanted to say for an overview is here. Okay, yes. So we talked about this yesterday in discussion, but I think it's worthwhile mentioning now. Is we get so zoomed into this process that we forget to take a step back and see how this is relatable to us. So when you eat, the foods that you eat, hopefully, are complex carbohydrates, not processed, complex carbohydrates, proteins, yada, yada. So the carbohydrates that you eat, they're not going to be in forms of single molecules of glucose. They are going to come in different shapes and different structures. So the first thing that you have to do is eat, and then that food goes to your gut, right, to your belly. And in your gut, you have enzymes, digestive enzymes. And these enzymes are going to break apart those covalent bonds, thus taking big complex structures and breaking them into individual glucose molecules or something very similar. So essentially, the enzymes in your gut are going to hydrolyze, so a bunch of hydrolysis reactions, and they're going to take polysaccharides and they're going to break them down via water, which is why water is so important when you eat food, because that's what helps digestion. So polysaccharides get turned into or modified into or broken down into monosaccharides by special enzymes in a hydrolysis reaction, which means you need water. Again, water is very important. Okay, once you have your monosaccharide, one example of a monosaccharide is glucose. This is the glucose that then can start, well, first it has to go through the blood. And so we talked about glucose transport. So it has to get into the cell, right? So glucose has to be transported. Okay. And so during different steps of digestion, it uses passive transport, and then other times it uses active transport. And the details there, are important, but I don't want to spend too much time in this class going over them, but we can talk about them in office hours if you want. But both types of transport are used so that glucose gets into the cell. Okay, so now it's in cells, and now glycolysis can occur. Okay, okay. Putting it all into perspective. Great. Let's see, anything else, anything else? I think that's good. And then once glycolysis is done, all 10 reactions, the final enzymatic product, okay, so whatever substrate number 10 was, got modified into the final product. So it's product number 10. Product number 10 is 2-pyruvate. So somewhere along the line, you now have 2 products. So remember when we started talking about enzymes, one of the things I said, like, yeah, sometimes an enzyme takes 1 molecule and makes it 2. So we're going to actually talk about an actual example of that in real life. Okay, so it's also important to understand exert and endergonic, or I guess review this. So in this slide here, what I'm showing you is this slide is a graph showing the loss or the need or the harvesting of energy. So here on the very far left, so it kind of goes through time. So you would start with a molecule of glucose on this side of the x-axis, and glucose goes through glycolysis. And then you can see the amount of time that each of these processes takes. So glycolysis, there's 10 reactions, so it takes probably a pretty long time. Pyruvate really is just one simple kind of 2-step process here, so that doesn't take too much time. And then citric acid cycle is eight reactions. And so what this is just indicating, I'm going to zoom in here. Here's your starting molecule, and if you can see that, you can't, so I'm going to do it. Here's your hexagonal, your hexose, your glucose, and each of these represents what chemical reaction. So this little stick right here, let's see if I can do this. This right here represents the arrow from substrate to product. Okay. And so what this tells you also with this graph, I like it. I know it seems confusing now or overwhelming now, but I find it really helpful, is that also at reaction number 1, a molecule of ATP was needed. At reaction number 3, a molecule of ATP is needed. And so you're saying, wait, they're using the energy. Yeah, because remember, negative delta G is exergonic, which means energy releasing. Positive, or moving up the y-axis, say up, means the reaction was endergonic and required energy. So if you move down the y-axis, it's a negative delta G and it's exergonic. So you can see that these things are moving up the y-axis, which is why reactions 1 through 10, or 1 through 5, we say are exergonic, because we started here, and at the end of reaction 5, we've moved up the y-axis. And then from reaction 6 to 10, there's a giant drop. Like if you just look at the overall start and stop point, huge drop, which means it went down the y-axis, which means collectively steps 6 through 10 are exergonic, with the tiny little exception of step or reaction 8. You can see a tiny little increase in or need for energy. Okay, again, this also tells you that it's at reaction 6, NADH is made, at reaction 7, ATP is made, etc. So this slide or this graph will appear a couple times throughout the next few lectures. And so I think over time you'll get used to what it means and how to read it. And so at the very end of glycolysis, you have your final enzymatic product, which is pyruvate, which happens to be the first enzymatic substrate for the next process. Okay? And down here, this tells you for every 1 molecule of glucose, and think about it, when you eat, you're not just Like if you eat a big bowl of spaghetti and meatballs, you're not just eating one glucose molecule, you're eating thousands of glucose molecules. So once your enzymes break that into individual glucose, those glucose, each glucose yields, this is a byproduct, so that's not energy, but each glucose will yield 10 NADHs, 2 FADHs, and 4 ATPs. And you're still saying, like, that's not that much. But remember, 1 NADH is equivalent to 2 to 3 ATPs. FADH, 1.5 to 2 ATPs. So these will eventually be converted into more ATPs. And that's just for one molecule of glucose. It's pretty wild, if you ask me. So let's see. Okay, so we just talked about that. What are NADH and FADH used for? So this is going to be in the electron transport chain and ATP synthesis. These are the power. So this is where they're used. How? They provide energy. How do they provide energy? In the form of electrons. We'll talk about that later, but just so you have it in the back of your head. Okay. Alrighty, so let's get started. Reaction number 1. So what this lecture is basically going to be about, and we're not going to go through each reaction in detail because I want you to have some practice going through some of this on your own and trying to spot how the substrate has changed. But I want to give you some examples of how to do that properly. So here is reaction number 1. Here is your molecule of glucose. Okay. And we know that it's an alpha glucose. Why do we know that? Because here's carbon number 1. The hydroxyl is below the plane. Okay, so you have to know how to number your carbons. And so alpha D glucose, you're going to have your aldehyde as carbon number 6. 5, 4, 3, 2, and then your carbon number 1. This was the carbon that was involved in a 1,4-glycosidic linkage, but our cool enzymes cut that. And so now we have a single glucose. Okay, so what I really like about the textbook is it highlights the places that you need to put your eyes, okay? Just like when I say, like, please use your cursor so we know where to look on the slide. So I'm gonna do that too. This is highlighted in blue. Why? Because this is the region that has been modified. So you don't really have to worry about too much more. So you say, okay, cool. The textbook is telling me to look at that hydroxyl group. And now I'm going to look at the product and I'm going to see what has happened. And you're going to see like, oh, wow, the hydrogen was popped off and instead there's a phosphate group there. Oh, cool. Now your thought process should be, wait a second. Where did that phosphate come from? Because I know science is magical, but it didn't just come out of thin air. So you have to ask yourself, where did that phosphate come from? And then you look at, well, okay, well, how did this happen? Obviously, there's an enzyme involved. That enzyme's hexokinase. Then you start picking apart that word. You say, okay, well, I know the word kinase has something to do with phosphorylating things. And then you look at this part. You're like, hexo. Okay, I know glucose is a hexose. Oh, I get it. So this enzyme phosphorylates a hexose. The hexose in question happens to be glucose, and that's correct. So how did this happen? So hexokinase, we say hexokinase, and I'm only going to write this out for 1 or 2 reactions fully because we'll be here for 2 hours if I wrote every single reaction out from start to finish. So hexokinase, and here's the important part, catalyzes that is the proper term for how an enzyme functions, right? We always, when I first asked you, what do enzymes do? Catalyze chemical reactions. Okay, cool. Now use that definition that you have. So hexokinase catalyzes the, what does it catalyze? Basically, what is it doing? It is catalyzing the phosphorylation of some substrate. So this is always the enzyme. You always start with the enzyme name. And then this phrase really shouldn't change. And then this is some activity, okay? And you always want to describe the substrate being modified, right? So it's catalyzing the phosphorylation of some substrate. The substrate in this reaction is glucose. Okay, so here's your substrate. And you need a little bit more detail. So the phosphorylation, great, but now I'm going to ask where, because there's a lot of places that molecule can be phosphorylated, and this picture tells you exactly where. So it's doing this at, and the way that we describe sugars being modified, we describe them by the carbon that's being modified, even though, there's my cursor, even though the phosphate is not directly connected to this carbon, this is the carbon that has been modified, carbon number 6. So we say that the phosphorylation of glucose at carbon 6, okay? So we'll just say that this is maybe a little more detail, with a where detail. And then why? Why did all this happen? To make, to generate, to synthesize, okay? The product. What is the product? Glucose 6 phosphate. Okay. And so you might be looking at this reaction above and saying, yeah, there's a lot of other stuff that are there we didn't talk about. Now's what we're going to talk about it. So in order to do phosphorylation, you need a phosphate group. So it just requires another sentence. Okay? So you can say the same enzyme, right? Something like to do so, hexokinase. It requires the cofactor ATP, right? We talked about ATP and nucleotides being cofactors, specifically coenzymes. And so what do these nucleotides bring? The ATP is bringing a gift to the enzyme. Hey, enzyme, here, I have 3 phosphates. You need one? Okay, here you go. And you need energy to do all this hard work? Okay, guess what? When you break off one of my phosphate groups, breaking that bond, you're going to get tons of energy. So go for it. And so the enzyme does that. So in order to phosphorylate glucose, hexokinase first has to hydrolyze ATP. Hydrolyzing ATP means chopping off a phosphate group. When you chop off a phosphate group, you break a phospho and hydride bond, and you release a bunch of energy, and you release the phosphate group that you need. And the result is now ATP doesn't have 3 phosphates, so now we call it ADP. And ADP is like, cool, I'll just go back into the cytosol, and you know, I still have 2 phosphates. I could be used for other things, no worries. So in this case, ATP is the cofactor for the enzyme. Okay, so that's pretty much it. That's reaction number 1 already. So the last thing that I want to say before we move on to reaction number 1, and I ran out of room, which is annoying. So let me just put it in a different color. I know what I'm going to do. Now I have space. Okay, so the last thing, when we started talking about enzymes, we also started talking about how enzyme regulation is critical for cells, right? Because we don't want an enzyme to be functioning when we don't actually need ATP. Like, we're good, we have tons of it. It's like when your gas tank is full, you can't keep putting gas in it because then your car does not like that. So same thing with ATP. When the cell has enough ATP, it doesn't need to do all these reactions because all of these take energy, like some sort of energy. So too much is not good. Not enough, it's not good. So we need to make sure that we address the enzyme regulatory steps. And so reaction 1 is an enzyme, so hexokinase. does get regulated. Okay? And it's a pretty stringent regulation, meaning that I don't know what the sensor is, but when you have reached these certain thresholds, this enzyme is one of the key enzymes that gets regulated. So if you think about it, this whole entire process of glycolysis and cell respiration is so that the cell makes more ATP. If the cell has enough ATP, we don't need to do this. If the cell does not have enough ATP, we need to do this. So why wouldn't a cell have enough ATP? Well, this reaction really just shows you one example of how a cell uses ATP. Hey, it uses ATP to actually do this stuff, and now we have ADP. So in this one reaction, you lost an ATP and now you have an ADP. So maybe when you have all your ATP, it's all like full tank of ATP, it's because you have phosphorylated every possible ADP. And so the number of ADPs and AMPs is really low. Every time you use an ATP, essentially you're making an ADP. You use another ATP, you've created another ADP. And so over time, this balance shifts to the point now where the cell really doesn't have very many ATPs anymore. It does have a lot of ADPs. So the cell's like, I know how I can make more ATPs. I just got to add phosphate groups to all these ADPs, and then I'll have more ATPs. So it's this balance between ATP and the hydrolyzed versions. So when ATP concentrations are high, this would you could predict that in the cell, ADP and AMP concentrations are low, and vice versa. When ATP concentrations are low, that is because you have hydrolyzed all the ATPs into either ADPs as seen right here or AMPs. Okay. Okay, so having known that, then we can say like, okay, cool. So when ATP is low, or when these things are high, we need to speed this process up because we need to make more ATP. And if you think about the flip side of that coin, when these things are really low, it's probably because the cell has a lot of ATP in its really rich phosphate source. So we don't need to do glycolysis, pyruvate oxidation, and waste our time and energy doing all that stuff. And so these molecules actually serve as enzyme regulators. So, I lost my train of thought. So this enzyme, yes, okay. So they serve to allosterically regulate the enzyme. So these, when the concentration of these are really high, they will go and bind to hexokinase, and they will change the active site, the conformation of the active site, such that glucose cannot bind anymore. If glucose can't bind, then it will never become glucose 6-phosphate and will not get to step 2 or reaction 2. So how does this get regulated? Regulation. It can be allosterically negatively regulated by ADP. So inhibition or negative regulation, the symbol for it is a sideways T. I don't know why, okay? When you regulate something and increase the activity, the symbol is an arrow, okay? So it's either an arrow or a flat arrow, like dead end, no more. Okay, so ADP and AMP can regulate hexokinase. And then all the same terminologies used. When it binds to the secondary site, conformational changes induced, the affinity for the substrate glucose is decreased, glucose does not bind, the product glucose 6-phosphate is not made. Okay? The other thing that negatively regulates hexokinase is when this product itself, when there's too much glucose 6-phosphate in the cell, this some of it goes on to reaction 2 and 3 and 4 and 5, but some of it comes back and binds hexokinase. So remember, a downstream product going back to inhibit an earlier enzyme, yes, it's at one step, but if this product is going back to inhibit an earlier enzyme, this is an example of negative feedback, right? So not only does ADP and AMP negatively regulate hexokinase allosterically, you also have G6P. Okay, G6P also negatively regulates this enzyme. This is an example of negative feedback. ADP is not an example of negative feedback because this is not a product. This is the product. This is a byproduct. Okay. So we don't say ADP negatively regulates the enzyme via negative feedback loop. No, this is a negative feedback loop. Oh. Okay. Okay, so any questions so far? We've only done one reaction. Okay. Okay, so reaction 2. And so what I'm going to say here is the titles for these slides, you'll see that I use the abbreviations for them. But the pictures that you see here have the full names, and those are the names that you need to know. And so what you'll also notice is that the product from this reaction is the substrate for the next reaction. So when we're talking about those big synthetic or metabolic pathways, this is a metabolic pathways where the product of the previous reaction becomes the substrate for the next reaction. And that just, it's a domino effect, just keeps on happening. So here's your glucose 6-phosphate. Here's the area that you have to look for or look at. And so what you can notice is the first thing that happens is that the substrate opens up into the chain form. And then after that, things start changing. So this is a big change, goes from ring form to open chain form. This is all catalyzed by this enzyme phosphoglucose isomerase. Okay, so now I'm going to start shorthanding it. And so, so that we can get through these in less than 20 minutes per reaction. Okay, so the enzyme phosphoisomerase, again, if you look at the name of the enzyme, it tells you a little bit about what it does and who it does it to. So let's look. Isomerase, so you know that it's an enzyme for sure. Isomer, you know that means changing the form of something. And then who it's changing the form of who? And so it's changing the form of a phosphoglucose. I would say this is a phosphoglucose, right? It's a glucose with a phosphate group on it. So somehow it's changing the structure of glucose 6-phosphate. How? So if you look at it, you can follow this. So it turns to straight chain, and then the chain really has some reconfigurations here, and all the minor details, no, they're not minor. Let's just call them details. All of the specific details about how these change from this to this, that's not of interest to me for this class. I promise if you ever take biochem in a university setting, you will be needing to do all these details. So we'll save that for that, professor. So really, I'm just interested in you being able to describe how the substrate, glucose 6-phosphate, is now fructose 6-phosphate. Okay, so phosphoglucose isomerase catalyzes the, okay, that never changes. And you can say the isomerization, you can say it catalyzes the opening of the ring form to the open chain, whatever you want. but I'm just going to write isomerization of, now the substrate comes here, the substrate name of glucose 6 phosphate to its open chain form. So it does two things. It not only, no, that's all it does, cool. So it opens it up, oh, and then it recloses it, right? To its open chain form, and continue. Restructures it to a 5-sided ring to make the product. The product is fructose 6 phosphate, and you would write that out. Okay. And so remember, fructose is still a hexose. You have not lost the number of carbons. Okay, you've just changed the shape of the ring from 6-sided to 5-sided. And that's the major difference here, is that you started with a 6-sided ring. In order to become a 5-sided ring, you had to open up the chain and then reclose it into a different shape. Okay, but no additional functional groups were added. The phosphate group, if you followed that, is still on carbon number 6. Nothing has changed, it's still there, okay? And that's it. No additional cofactors were needed. Boom, done. Okay, that's it. And this enzyme, so the fact that it has forward and backward arrows, as you might remember from chemistry, means that the reaction can go in both ways. And this enzyme here is not heavily regulated. So you do kind of see some back and forth in the cell happening at reaction number 2. Why? Because this is a reaction the cell wants to reach equilibrium. Okay. All right, so reaction number 3. The substrate from the previous reaction is now the product from the previous reaction is now the substrate for the next reaction. So here we have fructose 6 phosphate, here it is. Here's the area that we're supposed to pay attention to. And so the enzyme here is phosphofructokinase. So for sake of tongue twister, I'm gonna just call it PFK, but you do definitely need to know the names of the enzymes completely. So what does phosphofructokinase do? Well, if we look at the area that the book is telling us to look at, again, something similar that we kind of already saw. No longer do you have an OH, but instead you have a phosphate group there. Oh, wow, so now this thing has 2 phosphate groups. And if you look at the numbering system over here, let's take this numbering system and move it to the next slide so we know what each carbon is. So here's carbon 1, 2, 3, 4, 5, 6. Carbon 6 already had a phosphate group. Now carbon 1, here's carbon 1. Carbon 1 has the phosphate group. So essentially you would copy and paste all the stuff that you said about hexokinase, and you would just change some stuff. So if I took this, okay, let's see if it works. Put it here, great. Awesome. So you got to put the right words in there. Okay. Okay. So let's see if we can fill in the blanks using this template. I think that we pretty much can. So the enzyme PFK catalyzes in this case, the phosphorylation of what is the substrate? F6P at, it's not carbon number 6, but carbon number 1, to make fructose. We say bisphosphate by like 2. So fructose 1,6-bisphosphate. Okay. To do so, PFK hydrolyzes ATP into ADP. Thus, ATP is a cofactor for this enzyme. Boom, done. And so this is the second ATP that was used. So now we come up here, we use in reaction number 1, we used 1 ATP. In reaction number 3, we used 1 ATP. So right now we are in the deficit minus 2 ATP. What did I say? Reactions 1 through 5, you use 2 ATPs. There, we've used them. Okay. Oh, let's see, there's a quick question. The term phosphohexose is acceptable too? Phosphohexose for what? Not for the enzyme, that's not an enzyme name because it ends in OSC. So Francisco, what do you mean? Is phosphohexose acceptable for what? Okay, we'll come back to you. Okay, so what did I say though? We have to make sure that we talk about the enzymes that are heavily or majorly regulated, like regulation of them is very important. And so PFK is the biggest enzyme that regulation of PFK is like the most stringent, the tightest, like there's no messing up here. And I'll tell you why. If you finish reaction 3, the ability for the cell to stop any further reaction is really, really weak. So at this point, the cell really needs to take inventory about their ATP levels. If they need to make ATP, phosphofructokinase will be actively regulated. If they have enough ATP, phosphofructokinase will be negatively regulated. So PFK, regulation. Okay, so the first hexokinase we were talking about getting regulated just negatively. This one has both positive and negative regulation. So in this case, let's talk about the positive regulation first. And this is going to be via allosteric regulation. And the way that PFK gets allosterically regulated is when ADP is high and AMP is high. Remember, because if ADP and AMP are high, that means ATP is low because we've used a bunch of ATPs. We've hydrolyzed a bunch of ATPs. So these can come over to PFK, sit in their allosteric site, change the conformation of the active site to favor the affinity between PFK and F6P. And that will allow the enzyme to increase its activity and make fructose bisphosphate at a higher rate. Okay, negative regulation happens in a couple different ways, kind of happens opposite. So allosteric regulation again, when ATP is high. So too much ATP, cool. The cell will keep some of it to do all the, ATP jobs in the cell, mitosis, division, all these things, right? But if there's too much ATP, the cell's like, all right, ATP, can you go just make it stop just for a little bit? And so ATP can also negatively allosterically inhibit PFK. And that is the crazy part because ATP is also needed as a cofactor. So ATP has double jobs. It depends on the levels of ATP in the cell. Not enough ATP in the cell, the ATP is used to make phosphofructokinase. It allows it to do its job. Too much ATP in the cell, some of that ATP is like, I'll show you, I'm going to sit on you and not let you make more FBP. Okay, and then the other thing that negatively regulates PFK is a molecule called citrate. Okay, so citrate, does it sound like citric to you? I think it does. This happens to be the first product of the citric acid cycle. So this is product number 1 of the citric acid cycle. If this molecule exists in too high a concentration, the citrate, it's like, I'm a product, I better go back and negatively regulate an enzyme. So what is that an example of? This is negative feedback loop, right? A downstream product coming back to inhibit or activate, but in this case, inhibit an earlier enzyme, negative feedback loop, okay? Because it's a downstream product of a downstream process, and downstream just means like it happens later. All right. Okay. Cool. And you'll read in the in some of the resources that you can use the same rationale for levels of NADH and NAD+. So I'm going to kind of put that over here to keep that in mind. So remember, so here's the I guess the rationale. So NADH can be used it's the energy that will eventually allow the cell to make 2 to 3 ATPs. Okay. When NADH is used to do that, it actually becomes NAD+. Okay, so it's that electron from the hydrogen that actually powers the cell and allows it to make those ATPs. So when NADH is high, we don't need to make more NADH because we already have it. So when NADH is high, it typically inhibits enzymes. And then when NAD plus is high, that's probably because we've used all the NADHs, and that's why we have a bunch of NAD pluses. So NAD plus being high, this typically activates or promotes or upregulates enzymatic function. Okay, great. So reaction 4, this is when it gets interesting. Remember how at the very end we have 2 pyruvates? Well, this is going to explain why. All right, so the product from the previous reaction, FBP, is modified to become 2 products. One product is called dihydroxyacetone phosphate, or DAP. The other product is called glyceraldehyde 3-phosphate, or G3P. So how did this happen? If you look and you remember fructose is a 6-carbon sugar. It's a hexose. If you look at DAP, here's DAP. How many carbons does it have? 1, 2, 3. So there's 3 of the carbons. How many carbons does glyceraldehyde 3-phosphate have? 1, 2, 3. So essentially, let's look at this substrate really quickly. I'll say that this is a 3-carbon product. This is a 3-carbon product. Basically, the fructose ring was opened up, not magically. Aldolase catalyzed the opening of the ring into the open chain form. Once it was open, then aldolase also cleaved or cut or hydrolyzed the bond between the central carbons to create 2 products. Okay, and so that's essentially what happened here. No extra phosphate groups were added or removed. So aldolase is not a heavily regulated enzyme. It doesn't require an extra phosphate group. It does use energy from the cell to do this, though. Anything in the cell that happens, you're using cell energy. Okay, so I'm just going to highlight some of the things that the enzyme catalyzed. The enzyme catalyzed the opening of the ring of what? Of FBP to the open chain. And then it cleaved or cut or hydrolyzed the covalent bond between carbon 2 and carbon 3. Okay. And that's essentially just what happened. And then this to make DAP and G3P. Okay. So importantly, you're going to say like, cool, I'm getting it, right? So these products become the substrate for the next reaction. Yes, but only for one. only one of these products gets used in the next reaction. And the product that gets used in the next reaction is, I don't want to do that, is DAP. This is, so yeah, actually I do want to do that. This is the product that gets used as a substrate in reaction 5. Okay, so let's go look at that. And oh my gosh, what does it become? It becomes G3P. So up here, reaction 4 made a G3P. And in reaction 5, the cell converted DAP into a second molecule of G3P. And how did it do that? So let's look at the blue region here. Here's your DAP. The enzyme that catalyzed the modification is called triose. Why? Well, let's see. Triose phosphate isomerase. So what does the enzyme do? Isomerizing, changing the structure. Who is it changing the structure of? It's changing it of some triose phosphate. This is a triose phosphate because there's 1, 2, 3 tri. It's a sugar still. So it's a triose. So this is a 3-carbon sugar. Phosphate because it's phosphorylated. So this is essentially just changing the structure around. Nothing was gained, nothing was lost, just bonds are broken and reshifted. The molecular formula is the same, and it changed it from a DAP to a G3P. So at the end of reaction 5, you have no more DAP molecules, and you have 2 G3P molecules. So now from this point forward, every single thing that happens to one of the molecules also happens to the second one. We're not like putting it aside, we're not storing it for later. So basically you've been kind of traveling along this linear reactions. Now it splits and we have 2 linear reactions. The same reactions are happening for each molecule. Okay, so that concludes, so the end of reaction 5 is the end of the endergonic or positive delta G reactions. All right, any questions on reactions 1 through 5? All righty, cool. So now we're gonna start seeing how the cell is going to pay back the investment that it made and then start making money or making energy molecules. Okay, so you have 2 G3P. So this reaction cartoon only shows 1, but this is happening twice. Okay. So G3P gets converted to 1,3-bisphosphoglycerate. Mouthfuls. Okay, and so the enzyme that does this is called glyceraldehyde 3-phosphate dehydrogenase. And so the dehydrogenase part tells you what the enzyme does. The first part of the enzyme name tells you who it's doing it to. And now you're going to see something that you haven't seen before. You're going to see NAD being used. So obviously we know NAD plus is going to be something that the cell can use later. So, but with regard to the enzyme, this is a cofactor for NAD plus. So let's see what happened to G3P. So it's telling you to look at this hydrogen here. And this is carbon number 1, 1, 2, 3. Okay. And we know that because 3-phosphate indicates that carbon 3 is the one that has the phosphate, 3-phosphate. So this is carbon 1, 2, 3. If we look at the product, carbon 1, instead of having an H there, dehydrogen, removing the H, now it has a phosphate group. So now you're starting to get in the habit of saying, like, where did that phosphate group come from? Don't just assume that it came from ATP. Chances are it probably did, but sometimes it doesn't. And this is one of those sometimes. So this phosphate group, remember where we're at, we're in the cytosol. The cytosol is not just water, it has ions, it has phosphate ions, it just has a bunch of stuff floating around. And so this actually was recruited from the cytosol and used. So no ATP hydrolysis occurred at this step. So in this case, the enzyme, So now I'm just going to abbreviate it, but you should actually write the enzyme name. The enzyme catalyzes the, I guess in essence, if you're removing a hydrogen, you're essentially oxidizing it, but you can just say the removal of a hydrogen where, let's not put where yet. A removal of a hydrogen and phosphorylation. Now we can say at carbon number 1. Okay. To make the product, whatever the product's name is, bisphosphoglycerate or 1,3-bisphosphoglycerate is correct too. Okay, it doesn't do it magically to do so. Yeah, this is actually, I'm gonna actually use correct terminology here. Why not? I would want you to do that. So let's look at what's happening here. When you remove the hydrogen has to go somewhere. It doesn't, sometimes it just goes into the cytosol, but sometimes it gets put, most times it gets put somewhere else. And that's the whole reason that the enzyme needed NAD+ as a cofactor. So when the enzyme removed this hydrogen, it needed to put it somewhere. So it actually put it onto NAD+. So removing the hydrogen here, so when you remove it, by definition, you're removing an electron, right? So yes, I know a hydrogen has a proton and an electron, but you're removing an electron from the substrate. In essence, you are oxidizing. Oxidizing means removal or loss of an electron. So the substrate gets oxidized. So glyceraldehyde 3-phosphate dehydrogenase actually catalyzes the oxidation of G3P. And so if you're going to remove a hydrogen, you got to put it somewhere. So it's going to remove it and it's going to add the hydrogen here. When you gain an electron, you say that you are reducing this. Okay? So not only did the enzyme catalyze the oxidation of the substrate, oxidation and reduction are always paired. So that means you have to state what was reduced. The thing that was reduced was NAD+. Okay, and so the products or the effects of this, at least this part here, is to generate this molecule of NADH. That's really important. That's a huge, high energy storing molecule. And that is one of the goals, right? We want to make NADH because we want to be able to make more ATPs down the line. Okay, so not only did the enzyme catalyze the redox reaction, and you've described the redox reaction, but then it also catalyzed, so this has to happen first. You have to remove the hydrogen first before you put the phosphate group on. So this happens, and you're going to write it in chronological order. The redox happened first, and then it also catalyzed the phosphorylation, and all of this is happening at carbon number 1. And at the end of this, after you've oxidized and phosphorylated carbon number 1, you make the product 1,3-bisphosphoglycerate. Okay. And so when you talk about the phosphorylation, you also in your description want to say that the inorganic phosphate was from the cytosol. Okay. Yes. Cool. And so this is our first reaction where we're talking about redox. And so I just want to remind everybody that oxidation is loss of an electron. Okay? So, and reduction, you know, it's kind of weird, right? Because you like reduce, you're like reducing the number. No, it's actually not. Okay, so reduction is gaining an electron. Okay. So there's some mnemonics for this. So Leo Losing an electron is oxidizing. Leo's like a lion, right? And lions go grr, it's dumb, but gain an electron is reducing. So Leo the lion goes grr. And so if you can remember those things, then hopefully you can remember what oxidize means and reduction means. And when we're talking about cell respiration and making ATP, the electron source is actually the electrons typically from a hydrogen atom. Okay. And so that's why over here you can see that there is a proton that was released into the cytosol. Why? Because only the electron was transferred to NAD+, and the remaining proton was put into the cytosol. Okay. Okay, so this is really important because we're going to see NAD plus and NADH, we're going to see that used as a cofactor a lot of times throughout, maybe not any more times throughout glycolysis, but definitely in the citric acid cycle. Okay, and so I just want to show you, because everyone, no, not everyone, every semester someone always asks about NAD and what exactly is happening. And so what this shows you is on this side, you have your NAD plus molecule. So you see this nitrogen is positively charged and this hydride. So the electron from a hydrogen comes from somewhere. So in the example that we just saw, the electron came from G3P. So only the electron comes and attacks this, and then these bonds attack one another, domino effect, okay? And then the result is a reduced, so this is reduced, a reduced NADH. This is oxidized. And you go from NAD plus to NADH, and this can happen in both directions. And it's important to remind you that NAD plus and NADH are dinucleotides. Okay, so here it is in the oxidized form. So there's no electron, there's one less electron, which is why it's positively charged. And how do we know that it's a dinucleotide? Well, if we think about what a nucleotide is, a nucleotide is going to be a sugar molecule plus a base plus 1 to 3 phosphate groups. Okay, and so let's see, do we have that here? Here's one nucleotide, here's the sugar, here's the base, here's the 1 phosphate group. Okay, sorry, 2, oh my gosh, 2 phosphate groups. So this thing that I outlined is actually the same structure as ADP. And so here's the second nucleotide. So you have your sugar, you have your base, and I guess like in general, each of these nucleotides has a phosphate group. So this phosphate group belongs to this nucleotide, this phosphate group belongs to this nucleotide, and when they interact together, they form what's called NAD, which is nicotinamide adenine dinucleotide. And in the reduced form, you see one extra little hydrogen, but this has lost its positive charge. Okay, so this is the small, tiny little change makes it not energy storing to like, yes, this is the big money. Okay, so keep that in mind, that nucleotides are not just building blocks of DNA. Nucleotides are used for a lot of different things. Okay, reaction 7. No. Okay, so reaction 7, let's look at the part in blue. This is what's going to be changing, and it's catalyzed by phosphoglycerate kinase. Anytime you see the word kinase, you know that that is going to include, or it's going to be some kind of phosphorylation event that's happening. So let's see what happens. And in this case, we see that, all right, here's the blue section. If we look at the product, oh man, the P went away. Where did it go? It didn't magically disappear. Did it go into the cytosol? No, because this reaction says to us that ADP actually gets phosphorylated, okay? So when you phosphorylate ADP, you're adding a third phosphate, you have made ATP. So this is our first reaction here where we're making ATP, cool. And remember, this happens twice because we don't just have one BP. BPG, we have two of them. So yes, one reaction makes ATP, but this happens twice. So we're actually making our 2 ATP. We've paid ourselves back. The cell has paid itself back. Okay, so at this point, we have a net of 0 ATPs. Not great, but better than -2. Okay, so, you know, just quickly talking about this, phosphoglycerate kinase catalyzes the dephosphorylation of 1,3-bisphosphoglycerate at carbon number 1, right? So 1, 2, 3, right? And uses that phosphate group to phosphorylate ADP to make a molecule of ATP and the product 3-phosphoglycerate. So the enzyme catalyzes, what is it? And put it in chronological order. What has to happen first? You have to remove the phosphate, so the dephosphorylation of the substrate. And the phosphorylation of ADP, okay, to make the product 3-phosphoglycerate and a molecule of ATP. Okay. Alrighty. Cool. Perfect. Easy. This is the exception one. This is the one. So you can see here, if you're making ATP, this is definitely a negative delta G. If you're making NADH, this is definitely a negative delta G reaction, right? And this actually stores a ton more energy. And I think the word I have in my notes somewhere. Oh yeah, okay. So this reaction, anytime you make a molecule of NADH, this is about 100 kcals per mole. And the number isn't so necessary as is just comparing it to when you make a molecule of ATP. This is just about minus 25 kcals per mole. Okay, so reaction 6 is way more exergonic than reaction 7. And then reaction 8 uses a tiny bit of energy. So what happens in reaction 8? So let's look at this here. This is a dog hair that was on my book and I took a picture of it. So there's that. So 3-phosphoglycerate, let's look at the region in blue. Basically, it's just some restructuring, just like switching some stuff around from moving the phosphate group from carbon number 3 to carbon number 2. And in doing that, the amount of energy that was put in was a little more than any energy put out. So this is slightly, little tiny bit delta G, just a tiny bit. Okay, but same thing. Phosphoglycerate mutase catalyzes the, I don't know, restructuring of the phosphate group from carbon number 3 to carbon number 2. Okay, reaction number 9. So there's 2 enzyme names, not 2 enzymes involved. It's just you can find them in the literature 2 different ways. And so the book describes this as enolase because it turns the substrate into an enol, okay? But you will also see that it was phosphopyruvate hydratase. I use enolase, but I've seen this as well. So if you look at 2-phosphoglycerates, so 2PG, you look at this area that's in blue here, some things are happening where you actually, enolases, what they do is they kind of cleave the molecule, and then in doing so, they produce a molecule of water. So in general, they cleave molecules and produce, basically they cleave off an H, an H, and an O somewhere on the molecule to make a molecule of water, right? And so here's this part. There's the OH, and here's the extra H that make your H2O. Okay, and that's pretty much it from enolase. That's what enolase does. It cleaves off, or you can say hydrolyzes, or yeah, I think those two words are fine. Catalyzes the dehydration. So catalyzes, the activity here would be the dehydration of 2-PG, okay, to make phosphoenolpyruvate and a water molecule byproduct. No additional cofactors are needed here, no energy storing molecules are made here, and that's that. And this is slightly exergonic. Right? And then last but not least, we made it. Reaction 10. So the product from reaction 9, phosphoenolpyruvate, is the substrate for the final reaction. And so here we have our substrate, PEP, and here's our enzyme that is at play, so pyruvate kinase. What does pyruvate kinase do? Hopefully you're already starting to think something to do with pyruvate, something to do with phosphorylation, and now you're going to pick it apart. And so what you see is that the enzyme, let's look at the blue region. So, all right, cool, I see it. Oh man, all right, so the phosphate group is gone. Where did the phosphate group go? Oh, what else is in this reaction? We have ADP, and on the product side, we have ATP. Cool, I can see where this is going. So what happened here is the enzyme pyruvate kinase catalyzes the dephosphorylation. You gotta get the phosphate group first. So we always present in chronological order. Phosphorylation of what? Of the substrate. Okay. And the phosphorylation, where is it putting that phosphate group? It's putting it onto ADP, okay, to make pyruvate and a molecule of ATP. So this is the product, and this is an energy storing molecule. Okay. All right. And that's it. So you've made your pyruvates. And what I want you to see with the pyruvates is a couple things. If you look at this region right here, think back to your functional groups. This functional group is a carboxyl group, an ionized carboxyl group, right? It's your COO minus. And this right here, you don't know this functional group, but I'm going to tell you it right now. This is an acetyl group. Okay, there's your acetyl group. So pyruvate is basically two little functional groups attached together. And so why do I say that? Because there's important things here. Pyruvate is an ion. It has a charge. If it's a charge and it's in the cell, that means it's polar. Okay, so pyruvate is an ion. What kind of ion is it? If it is ionized, it is an anion. Okay. And if it's any kind of ion, it doesn't matter if it's anion or cation, that means this thing is polar. I would say this is a relatively small molecule. It's not made of a bunch of atoms, but it's a polar molecule. And remember where pyruvate has to go if we're in eukaryotes. All this was done, all the 10 steps of today were done in the cytosol, but pyruvate oxidation happens in the matrix. So now you have to get this negatively charged polar molecule into the matrix, which means you have to pass two membranes. Last time I checked, polar molecules don't just simply slip through the membrane. Why? Because they're polar and they cannot go past the hydrophobic tails. So we're going to keep that in mind. And so I just want to recap all of glycolysis right now. So you start with your glucose molecule. You have steps 1 through 5 where ATP was used at two separate reactions. And then at reaction 4, you now have 2 products. And then one of the products gets turned into G3P. So now you have 2 of everything. And so that's why you have 2 arrows here, because this is the start of when you have G3P. You have G3P here and G3P here. Okay. And so each of the G3P molecules go through all of reaction 6 through 10, like we just talked about. And at the very end, this G3P goes through all of this to make your pyruvate molecule. There's your carboxyl group, your acetyl group. Okay. And so these 2 molecules of pyruvate need to go into the matrix if you're a eukaryote. Take that in 1 second. So here's your pyruvate. Has to cross your outer membrane. So if we zoomed in, it would look like this. And then your inner membrane. This would also look like that. Your phospholipid bilayer. Because it's polar, it needs some kind of carrier channel, something kind of proteins in the membrane that is going to allow it to cross both of those membranes. Okay, because it needs to get in here, right? So I think that's where we're going to leave off, and we'll start there, whatever, Thursday. And so just remember that this part of the molecule makes it polar. Okay? Yeah. Cool. Yeah, that's fine. So we'll stop there. So thank you again for letting me start a couple minutes late. I appreciate that. Now I'll take any questions. I know that there was some chatter in the chat, but I'm thinking it was between you all. And if your question was not addressed by a peer, please ask now. Go ahead, Max.
My question relates to reaction 6. If you could scroll up by chance. So that H that reduces the NAD, does that come from the cytosol, since there's also an H as a product on the right?
It gets removed from here. So think about what a hydrogen is. So when this gets removed, there's 1 proton and 1 electron. So the electron is what gets added to the NAD. And then the proton part of it is what gets added into the cytosol.
Oh, okay. Okay, thank you.
Good?
Yeah, that makes sense.
Okay. All right, Ashley.
Hi, yeah, my question relates back to either reaction 4 or reaction 2. So when the substrate is modified in between the ring form and the open chain form, is there an enzyme that's required to make that happen?
Yep.
Okay, thank you.
The enzyme here. Yeah, and I think they just didn't want to put the enzyme name again here, but yes, this is all due to this aldolase. And you got to also remember that in bacteria and some special kinds of plants, it already exists in this chain form. So yeah, I think that's why they don't like make a huge deal about it. But yeah, if it's going from ring form to open chain, like if aldolase is the only enzyme at this step, aldolase is doing all of that.
Okay, thank you.
Sure. Any other questions?
One more on reaction 6. The enzyme in it sounds pretty specific. Does it only work on that one type of sugar?
Yeah, so this would be an example. Great eye. So this would be an example of a lock and key mechanism. Whereas something like hexokinase, that can probably bind not just glucose, it just happens to most commonly bind glucose, but there might be, maybe it can bind mannose and just change it just a little bit to convert it into fructose or glucose 6-phosphate. Yeah, so yes, very good. Okay, any other questions? Okay, so I know it was a lot of crazy words and a lot of reactions, and I get it. So the advice that I want to give you is to go over glycolysis as soon as you can. Like if you don't have lab today, I would do it now, reorganize your notes. And what you might want to start doing is, I'm not a flashcard advocate very often, not for this class, but I really do strongly feel that in order to grasp this material, this first step is flashcards. Or something like Quizlets. And so you can construct your flashcards in two different ways. You can put an enzyme name on the front of the flashcard, and on the back, all the steps that the enzyme does. Or you can have substrate names in the front of a flashcard and what happens to the substrate and how. And so once you learn it that way, and you'll be able to learn if I just give you an enzyme name, you'll know what reaction I'm talking about. If I just give you a substrate name, you'll know what reaction I'm talking about. And so sometimes students will have both stacks of flashcards, and they'll just make sure that they know both of them, forward and backwards. So I will never ask you things about reaction 6. So when you start talking about this, when you talk to me or ask me questions like in reaction 6, I'm going to say, I don't know what that means. Start with a name that's specific. So the number 6 means nothing to me. But if you said when G3P is a substrate, then I know what you're talking about. So you should also get out of the habit of trying to remember steps by numbers, because on the test, I will not say in reaction 5, I won't give you numbers. I will just say, how does the enzyme PFK get regulated? And you'll have to tell me. Or I might say, in the first reaction where ATP is used, that's your clue. Okay, so any more questions? Cool, so we'll pick up on pyruvate getting into the matrix and getting oxidized there on Thursday. Thank you.